Yes, sir, we are live. You can start, है ना?
Yes. Yeah. Hi team, good evening. Today we are going to discuss the very important topic and basic of each and every kind of roles. As like I said in earlier sessions, clinical trial knowledge is very essential and very required. Whatever the interview you are facing, whether it may be clinical research kind of roles like clinical research coordinator, clinical research associate, centralized monitoring assistant, or else like I said no different kind of software jobs a pharma or a health science graduate and postgraduate can do. What are those jobs? I said no yesterday in depth, like clinical research jobs, clinical data management, statistical analysis system associates, that SaaS jobs, and pharmacovigilance jobs. Whatever the interview you are going to attend, the basic questions, the basic knowledge will also be checked on clinical trial. Okay, clinical trials are such an important aspect which will determine which will be the deciding step to release the drug into the market, because if the drug is not at all proven safety, so after so many incidences we have to prove safety to FDA before the drug released into the market, like thalidomide disaster, Kepower amendment. After so many all this and the WHO international drug monitoring program collaboratively monitoring very effectively to prove the drug as safe before releasing into the market. How you will be proving the drug as safe? You do the clinical trial. Clinical trial is nothing but what is a clinical trial? According to your knowledge, what will do in clinical trial, team? Anybody?
Fun. After
Yes.
clinical trials, we will do on the first time on humans.
Yes. So your clinical trial is a process in which will determine safety and efficacy of a new drug. It may be a drug, it may be a vaccine, it may be a surgery device. So whatever that should be released into the market, initially you have to prove the drug or device or vaccine as a safer product. Post submitting all this information of drug safety to FDA, then only we'll get approval from FDA to release the drug into market. So this is also in clinical trial we'll perform the new drug. For new drug we'll perform the clinical trial and we'll test the drug on subjects. As well, we'll compare the effects of that new drug with already standard marketed drug, the drug which is already marketed and which is showing its actions and which is having good compliance to patient. With that standard reference drug, we will compare our investigational product. It is the same, right? Because in the lab also, if you are performing some analysis lab or pharmaceutics lab, if you are doing some tests or some evaluation parameters, immediately you will check the values with reference compound. Why? If the testing compound is showing more and better kind of results than the standard one, then only we'll approve that for further process. If that new drug or new investigational product is showing less effects and compared to the standard marketed release product, why we should release one more chemical moiety? Because the chemical moiety which is already existing is showing its good actions and it is safer compared to the new investigational product. So for that reason, the investigational product whatever we were testing in the clinical trial, it will be compared with the standard marketed product. So that marketed product, our investigational product should show more safety, more efficacy than standard marketed release product. So then only the FDA will also just will give a justifying reason to give patents. So that much important this clinical trial is in the process of drug delivery, drug release into the market. So let's see what are the phases of clinical trials. Team, how many phases are there in clinical trials?
Pages. 4 pages.
Four
Four.
phases.
So in four phases, we'll be testing on what? Animals or human subjects?
Human subjects.
So yes, by based on the PCT information, before clinical trial, we'll be conducting preclinical trial. From that preclinical trial, what is the information we'll get? General pharmacology and toxicology information. General pharmacology information in the sense like pharmacokinetic, pharmacodynamic information. Toxicology information means we come to know which organ is getting affected more. By that, we can take some precautionary measures. By that, we can have an idea to whom we have have to select, to whom have to reject. That information will be getting from preclinical trial. Post preclinical trial will be sending any documentation or any approval to the FDA. Do we need any approval to conduct clinical trial after PCT, or can we go as simultaneously after PCT and can we conduct clinical trial?
FDA
Approval
approval.
of
You should get approval from FDA before
FDA.
conducting.
What is
clinical
that
trials.
approval? What is that?
FDA approved.
FDA approval. What is the document you'll be sending? You will be sending all the PCT pre-formulation formulation information. No, what is that form you'll be sending to the FDA?
INDA.
Yes, that is investigational new drug
New
application.
drug
That
application.
will be containing all the information about PCT, pre-formulation, formulation, and all the toxicology, general pharmacology information that will be reviewed by the Food and Drug Administration, and post that will be approved to conduct phase 1 clinical trial. So phase 1 clinical trial is that in which human subjects were taken. So the phase 1 clinical trial are also called as human pharmacology trials or non-therapeutic trials, because these are only given to healthy volunteers. Okay, phase 1 is given only for healthy volunteers, not the human subjects here should be healthy volunteers, not patients. Phase 1 trials are designed to find out if the drug is safe rather than whether the drug is effective. For example, if the patient is having fever and we have to test the drug to decrease the figure, whether the drug is acting and reducing the fever or not, then we can come to the no efficacy parameter. That's it, right? If we are giving a drug to the patient who had fever, so whenever we were giving the drug should act and should reduce the body temperature. So it is giving some action. Then we can determine the efficacy of the drug. But you are giving an antipyretic drug to the human normal subject who is having normal body temperature. That 98 degrees is only the patient, the subject is having, but we were giving the drug. Then there is no point of curing and reducing temperature and all that. There is a point of safety estimation. If you are giving a drug to the patient means the person who is deceased. If we are giving a drug to the deceased, then the drug show its action. The drug will show its action and it will cure the disease. By that we can determine whether the drug is showing its action or not. So we can tell drug is effective or not. But here in phase 1 clinical trial, we'll be giving drug to the healthy volunteers without any disease. So here the goal of giving the drug to healthy volunteers is to determine the safety, whether the drug is causing any adverse drug reaction or not. So there is no matter of efficacy determination in the phase 1 trial. That is why these trials are called non-therapeutic trials, because there is no pharmacological action. We were estimating, we were only dealing here with the safety. After giving the drug to the healthy volunteer, if the healthy volunteer is experiencing some adverse drug reaction, then we'll be considering the drug as non-safer. Okay, so if the drug is not causing it any ADR at normal doses, then we can consider it as safe drug. So here that's why the phase 1 trial are also called human pharmacology trials or non-therapeutic trials. A phase 1 trial is the first step in testing a new drug in humans. Phase 1 trials are conducted to find a safe dosage. We just wanted to find safe dosage, whether it's effective or not, we don't mind, because we are not giving it to patients. We are giving it to healthy volunteers. Means there is no disease in the subject. They are very healthy. We were giving the drug, that new chemical moiety, into the subject to determine whether this drug is causing any adverse drug reactions, which are not intentional, which are not favorable to the body condition. That we can determine in the phase 1 trial. So, and also we can decide how the drug should be given, whether it by pill or by injection. We can come to know by phase 1 clinical trial, like what is the formulation is compliant to the subject. Compliant means which can be administered easily and which can be administered without any ideas, through which formulation it is giving ultimate through which formulation is not giving any side effects or adverse drug reaction. That formulation also we can have idea and we can confirm in this phase 1 trial itself. So we can determine the safe dosage, the dose at which we don't get any ADR, that can be determined in phase 1 trial, as well as we can determine what is the formulation we have to administer in further process. Next, to observe how the drug affects the human body. Drug affects the human body. It is pharmacodynamics, like how the drug is causing, whether it is causing any areas like that. So there is no point of treatment here because the human subject is healthy and volunteer. There is only point of areas, safety information we were evaluating. We are giving this drug and we are coming to know whether the patient is experiencing any subject is experiencing any nausea, vomiting, or any other unintentional activities or actions for the drug. So that kind of information only will get from the phase 1 clinical trial. Phase 1 trial generally involves small group of people, that is only 20 to 80. Okay, phase 1 trial involves only how many number of people? 20 to 80, and healthy volunteers. Here only healthy volunteers will be involved in the phase 1 clinical trial. Next, phase 1 trials are of two stages, that is single ascending dose studies and multiple ascending dose studies. Single ascending dose studies is that in which small group of patients are given small doses of the drug and observed for a specific period of time. So initially what they will do is there will be subjects now in the phase 1 clinical trial like 20 to 80 subjects. So they will be dividing into groups and the drug will be given in ascending doses like 2 mg, 4 mg like that. So initially for the first week, they will select a group and for that group, they will be giving a smaller dose. Okay. Small doses over a specific period. Maybe you can assume like 1 week of time. So for that 1 week of time, they will be giving one dose. Okay. In this patient, first week, we were selecting first group. No, for first group, we were giving one dose, which will be a smaller dose. Smaller dose. If you are not at all observed any adverse drug reaction during this period, then a higher dosage of the same drug, then the higher dosage. Now you have tried 2 mg for one group. No, next we'll try 4 mg to new group of individuals for the next week. Okay, for the first week you have given very smaller dose and you have observed for a speculated time. After that, more than the 2 mg, you have given one more dose for different group. Okay, this process continues until intolerable side effects start showing up. This stage denotes the maximum tolerated dose of the drug. I said no, during clinical trial only we will be coming to know at what dose the drug is showing actions and what dose the drug is safer. That dose will be fixed and that documentation will be sent to FDA through the format called NDA, new drug application. Then post review of FDA, that drug will be released into the market with that doses. So here only that phase 1 clinical trial only will come to know at what dose the drug is safer. So they will be, I said no, for the first week, they will be giving to one group of subjects a smaller dose. For the next week, they will select one more group and they will give one more dose, that is increased dose, 4 mg. Next third week, they will select one more group and they will give 6 mg. For fourth week, they will select one more new group and they will give 8 mg. For coming weeks, they will be selecting on new groups and increasing doses like 10 mg, 12 mg, 14 mg. They will increase the doses to the newer groups until the patients, the subjects are getting maximum tolerable side effects. It means intolerable at one stage. If you are going on increasing the dose at 20 mg, the patient will get severe adverse drug reactions, which cannot be tolerated by the subject. So that will be considering as intolerable side effects. The dose prior to the dose, the subject getting maximum side effects. So at what dose the subjects got severe adverse drug reaction? 20 mg. So before 20 mg, the subjects were treated with 18 mg. So the the dose at which the subjects got severe adverse drug reactions, prior to that dose will be taking that as maximum tolerable dose. Maximum tolerable dose is that the subject where he or she can tolerate the drug dosage and getting not severe adverse drug reactions, and the dosage at which the subject cannot tolerate the adverse drug reactions, the prior dose to that intolerable dose will be considering as maximum tolerable dose. Okay, that dose only will be fixing and will be releasing into the market. So guys, you got the idea about maximum tolerated days? Any doubt? Any doubt? So what is the maximum tolerated dose? Team?
The dose which can be tolerated by the subject, the
Yes.
maximum dose.
So the dose which can be tolerated by the subjects is called maximum tolerated dose. So before which we'll consider this maximum tolerated dose, we'll be giving a higher dose, the dose at which the subjects are intolerable to adverse drug reactions. So the point at which the subject is causing severe adverse drug reactions. Before to that dose, the subject is getting a good therapeutic action. The adverse drug reactions are also tolerable. But once you are escalating the dose, which is 20 mg, the subject got huge adverse drug reactions. So that adverse drug reactions which are intolerable to the subject body. So before to that intolerable dose, there will be one safer dose, no? That dose will be considering as maximum tolerable dose. Okay, that will be calling as safer dose. So that is the process involved in single dose studies. Hereby we can come to know what is a dose safer for the subject and which can be tolerated by the human body. That dose only will be fixed and formulated and released to the market. Next, multiple ascending dose studies. The main purpose of this phase is to evaluate the safety, tolerability, pharmacokinetics, and pharmacodynamics of a new drug on humans and the side effects associated with the increasing doses. What happens is in single dose ascending studies, what we did for the first 1 week, what we did, we selected one group of subjects and we have given 2 mg. For the next week, we have selected newer group and we have given one more dose, that is increasing doses. Simultaneously, we did the same until we are getting maximum tolerable dose. Unlike that, in multiple ascending dose studies, what happens is, in multiple ascending dose studies, the subject dose will be divided. Okay, for example, you consider 10 mg. So to the same patient in the given interval in the week, during a week of time, to the same patient, initially they will be given 2 mg. Okay, initially they will be given 2 mg, and next they will be giving 4 mg. The dose will be divided. The 10 mg drug formulation will be divided into 2 mg, 4 mg, 6 mg, 8 mg, 10 mg, like that. And to the same patient, all the doses will be given. Here, what happened in the single dose ascending studies? To the newer groups, we were giving newer escalated doses. But here, unlike that, to the same dose, there will be one group of subjects and we'll choose one patient. To the same patient, we'll be giving escalated doses, 2 mg, 4 mg, 8 mg, 10 mg. By that, we can come to know at which dose the patient got more adverse drug reactions. And also in the same patient, because there is a diversity, maybe one group of subjects maybe tolerate 20 mg, one group of subjects, that one person body may tolerate higher than 20 mg. There possibilities there, no, based on the resistance and tolerability of the human bodies, the drug intolerability also will differ. So for this reason, what happens is in single dose and ascending studies, to one group of subjects, they were giving one dose. To the next group, new group, they were giving new dose. But unlike that, in multiple dose ascending studies, for the same patient, the dose will be divided 2, 4, 6, 8, 10, like that. So to the same patient, the escalated doses will be given. To the same patient, will be determining different parameters. So that is the difference between the single ascending and multiple ascending dose. And the same pattern will be followed for the rest all groups also. So the dose will be divided and the dose divided will be given to the patient simultaneously. After one dose, remaining dose. After remaining dose, another dose. So like that, escalated doses will be divided doses will be given for same patient. There, different group of subjects. Here, same patient will be given the divided doses. Okay. By that, here we can come to know safety of the drug, tolerability. What is the tolerability by the patient, kinetics, dynamics of a new drug on humans. So these kind of information also will get from single ascending studies. Multiple ascending studies also will come to know. Okay, that is the difference between SAD and MAD. Okay. Next, phase 2 trials. Phase 2 trials are conducted in about 100 to 300 patients. What is the count of phase 1 trial? How many subjects will be involved?
20 to 80 members, 80
Yes.
patients.
So here the subject's number is so we don't have to call here patients. It was a mistake. So we don't have to call them as patients. We have to call them subjects. While in trial, we were treating some subjects. Then we have to call them subjects. So whether the count is increasing or decreasing? Increasing, right? Compared to phase 1, phase 2 trial count is increasing. There 20 to 80, but here 100 to 300 subjects. These are called therapeutic trials. What about phase 1? They are therapeutic or non-therapeutic? Phase 1 trials are therapeutic trials or non-therapeutic trials? Team?
Non-therapeutic.
Non-therapeutic trials.
Why non-therapeutic trials?
Right. To find a safe dosage.
Yes, we will be finding only safety parameters there. We are not at all dealing with the efficacy parameters. But in phase 2 trials, what happens is we were giving the drug to deceased subjects. Okay, deceased volunteers. So in phase 1, we'll be dealing with healthy volunteers, healthy human subjects who are not having any disease. Okay, in phase 2, we'll be dealing with deceased volunteers. Whenever we were giving a drug, a new chemical moiety to a deceased candidate, for example, take some antipyretic drug. So the patient, the subject got the subject we were taking here is pyrosis subject. Okay, the subject who is having higher temperature. So whenever we were giving this antipyretic tablet, what this antipyretic tablet will go and do? This antipyretic tablet will go inside the human body, that will reduce the temperature. So here we are giving the drug to the deceased candidate. For example, take paracetamol. So we were hiring the subjects, we are recruiting the subjects who are deceased, were having fever. So to the subjects, we were giving the drug, antipyretic drug, so-called paracetamol. So whenever we were giving fever, paracetamol to the fever patient, what happens? This paracetamol will go and act on that mechanism and it will reduce the temperature. Here, what is the difference between phase 1 and phase 2? There it is showing safety parameters because the subject is not at all having any abnormal body condition. But here the subject is having abnormal body condition, so it is going inside the body, the drug is going inside the body and showing its action means here we were determining efficacy. So meanwhile, while efficacy determination to the deceased candidates, whenever you are giving a drug which is going and showing its action, meanwhile there is also probability of getting safety ADRs, right? So even though it is reducing body temperature, it will sometimes sometimes not sometimes, most of the times it will give, like, because it is a foreign particle, it is a chemical moiety, whenever introduced into the human body, it will reduce the temperature, that is a fine, but it is also cause adverse drug reaction, maybe nausea, maybe vomiting, or some dehydration. Based on the drug, the ADR will be differs. So it is also giving some ADR. Meanwhile, parallelly, it is treating the disease. Meanwhile, it is also causing adverse drug reactions. So we'll be getting information like safety and efficacy information from phase 2 clinical trial. That is also called as therapeutic trials. Why we're calling therapeutic trials? Because in this phase 2 therapeutic trial, we'll be determining safety as well as efficacy parameters, because we were giving the drug to the deceased volunteers. Unlike phase 1 trials, there normal healthy volunteers will be administered the drug, but here unhealthy subjects will be given the drug. By that, the drug will show its action. So these are the goals of the phase 1, phase 2 clinical trial, to determine if the drug has an effect on a particular disease. That's what we were giving the drug to the disease candidates, right? So if it is really having action, then it will cure the disease. Then we can document the information. To observe how the drug affects the human body, that is dynamic information, as like trial 1. Next, phase 2 trials continue to test the safety of the new drug and begin to evaluate how well it works against a disease. So it is telling that we'll come to evaluate here safety as well as efficacy information. Phase 1 trial is only intended for safety evaluation. Phase 2 trial is intended for safety and efficacy determination, because here the subjects were deceased volunteers. Next, phase 3 trials. These are also called therapeutic trials. Here also we can determine the safety and efficacy determination. The same process, whatever you are doing in the phase 2, that will be done here. So why we should do the same trial again? Why we should do the same phase 2 process here again? Anybody? Any wild guess? So the phase 3 also therapeutic trials, and this will also be done on the deceased volunteers. Here also we'll come to know how the drug is. Yes. How the drug is safer, how the drug is treating the disease. Same kind of parameters will be evaluated in phase 3. What is the difference between phase 2 and phase 3? Anybody? Any wild guess? Why we should do the same process again?
Increasing subjects.
Yes, very well, sir. So compared to the phase 1, here 100 to 300. Here how many? 1000 to 3000. Here these are only therapeutic trials. Here these are therapeutic confirmatory trials. Why we are calling this as therapeutic confirmatory trials? Because during phase 2 clinical trial, we will be testing the drug only on how many subjects? How many subjects? 100 to 300. But here 1000 to 3000. After release of the drug into the market, it will go and expose to various kind of population, right? Nearly millions of people will be taking the drug. So it will be the maximum count will be 10,000 in the clinical trial. But once the drug released into the market, it will go worldwide. It will have chances of exposing to more and more millions of patients, right? So for that reason, if we don't like, it will be not sufficient. The documented information, whatever you are getting from the therapeutic trials, that is phase 2 trials, will not be sufficient. You should prove the same therapy safety and efficacy information on some more subjects too. Here we were confirming, here we were increasing the subject count, and here we were doing the same trial again on more number of subjects. Here we were determining again safety and efficacy, but the subject count is varied and increased when compared to the phase 2 trial. By that we can have some confirmation. So here we can have some more patient, some more subject information, some more subject data. By that we can confirm. So this drug is now exposed to more than nearly 300 subjects, and it is showing its safety, and it is a efficacy. It's showing its efficacy, and it is a safer product. We can confirm the product, because after phase 3 only we'll submit new drug application. New drug, if you want to submit new drug application, then FDA will review each and every point. Then the information whatever you are getting from phase 2 trial, that is only on limited number of subjects, that is not sufficient to approve the drug and give patents and release into the market. For that reason, we need confirmatory trials. Confirmatory trials are phase 3 trials. Phase 3 trials are nothing but therapeutic confirmatory trials. Phase 3 trial involves large group of individuals. Phase 3 trials focus on to know how a new treatment compares to standard or the most widely accepted treatment. I said no, during clinical trial we'll be comparing our new drug safety and efficacy parameters with the already widely accepted or widely used standard product. Next, to gain additional information regarding the effectiveness and the safety, to evaluate the overall benefit or risk ratio of the new drug for a large group of individuals. Compared to phase 2 trials, phase 3 trials will be having large group of subjects. We can come to know more information about benefit risk ratio. What is this benefit risk ratio? Benefit is nothing but how the drug is treating disease. Risk is nothing but what are the adverse drug reactions caused due to the drug. So the benefit risk ratio always should be justifiable, and it should be more enough. The information should be more enough to document and submit to the FDA. To compare the new drug with the current standard drug, that is what we do. I've already discussed. So now we got phase 1 information, phase 2 information, phase 3 information. Phase 1 information is about safety, that is on healthy human volunteers. That is a count about 20 to 80 volunteers. And phase 2 and phase 3 are therapeutic trials. The difference between phase 2 and phase 3 is confirmatory trials are phase 3. So total number of subjects in the phase 2 will be 100 to 300. Phase 3 will be 1000 to 3000. So now we'll be getting more cumulative data from phase 1, phase 2, phase 3. What is the information we got now? Safety information as well as efficacy information. Safety information, because whenever we were giving the drug, it may cause ADRs. That information will be documenting and calling as safety information. Now in phase 2 and phase 3, we'll be giving the drug to the deceased candidates. Then we can come to know how the drug is performing and how the drug is showing its action. Then we'll come to know safety and efficacy parameters and information. That all information will be documenting and submitting to FDA through a form called new drug application. New drug application. Unless you are not submitting new drug application, the FDA will never give approval to release the drug. We don't see phase 4, because phase 4 is that post-marketing surveillance where the drug will be released into the market. So the new drug application is a critical component in the drug approval process, which is required to submit to USFDA before the drug commercialization. Drug commercialization is nothing but getting marketing authorization for the drug. The sponsor company will be getting marketing authorization, then he will be called as marketing authorization holder. That is nothing but patents, approval, license to release the drug into the market. So post submission of NDA only, the FDA will review each and every point of the NDA and will go to the investigational sites and will check each and every process what they performed, and check each and every record what they have maintained during clinical trial, how they followed ethics, ICH, GCP, good clinical practices or not. All these things will reviewed by the FDA. Then only it will approve the drug and it will approve to release into the market. So the data gathered during the animal studies and human clinical trials become part of NDA. Although you have submitted the preclinical trial information through a INDA, you will be again submitting the cumulative data, preclinical trial data as well as clinical trial data through a document called NDA, that is new drug application. The goal of new drug application is to provide enough information to permit FDA review to reach safety, efficacy, and quality for pharmaceutical production. That is what I said. No? Here from the PCT and CT will be getting all the information regarding formulation information. What is the quality of the drug maintained, whether ethics are followed or not, what is the safety information, because here we are doing the trial on healthy volunteers and disease candidates. So we'll be coming to know what is the efficacy of the drug. All this information will be required for the FDA to review and approve the drug. So that information will be documenting and submitting to FDA through NDA. When it will be doing after the completion of phase 3. The NDA is filled with regulatory authority. After the completion of phase 3, we'll submitting the NDA. The new drug application must include what the drug is intended to, composition of the drug. So what is the drug intended to means? What is the purpose of giving the drug? For what reason you are giving the drug? For what indication? To treat for what body condition you are giving the drug. Next, composition of the drug. What is a polymer? What is the excipient? What is a all kind of different kinds will be there, no excipients, drug formulation. That all composition of the drug also should be documented. Manufacturing information. So how the drug is manufactured, clinical evidence for its safety and efficacy. That is nothing but clinical trial information. So evidence is nothing but we were proved the safety and efficacy in the human volunteers. That proved evidence should be documented and submitted and included in the new drug application. Adverse results, if any adverse drug reactions are causing, that is safety information. Controls and analysis. What are the controlling measures you took and what kind of analysis you performed? All this kind of information will become a part of new drug application, and this is a must and should criteria, and must and should mandate information should be included in NDA. So that after submitting the NDA, that regulatory authority, what they will do is the regulatory authority inspectors will visit clinical investigators to ensure that I said no, they will check each and every point of the NDA. Again, they will visit the investigational site, how the trial is performed, whether ethics are maintained or not, how the drug is given to the subjects, like who are the people involved there. So all this kind of information will be evaluated and cross-checked and inspected by the regulatory authority, FDA inspectors. The records are consistent with those provided in the NDA. During clinical trial, they will be documenting in the case report forms. In the coming sessions, I will be explaining in detail about case report forms, what is institutional ethical body, that is IRB, Institutional Review Board, Independent Ethics Committee. I will be taking care of everything, and you don't worry about that. So there will be maintaining of records during clinical trial. What the records they will maintain during during clinical trial, what is the process will be going on? They will be giving drug to the subjects, right? That subjects, after giving the drug, they will show some action. If the disease candidate is given, then we can evaluate how the drug is acting, whether the subject is getting adverse drug reaction and all that. So if the subject is getting adverse drug reaction, what kind of treatment you have given, everything should be documented in CRFs, case report forms. So there are so many records will be maintained during clinical trial. Even for the smaller lab practical only, we'll be having some log books, records and all that. So it is a huge process in the healthcare. So that is why for each and every process, even though you are giving drug, even the patient got ADR, even the patient getting good results, everything will be documented in records. So that documented information will be already submitting to through NDA. That is fine. Again, you have to maintain the records. So the CRF information will also be documented in India. That is fine. That will be submitting to FDA. But again, we have to maintain each, like if you are using any equipment or something else, that also we should maintain records. So that records will be cross-checked. So whatever the information is there in records, that is also should be there in NDA. So for that reason, when the inspection will goes on, the regulatory authority team will inspect the investigational site, that is clinical trial site, will cross check the records which were there in clinical trial with the NDA. So if the information is the same or not, any deviation, any manipulations like that, whether the patients have been adequately protected, that means ethics are maintained. The drug regulatory authority, after carefully reviewing the data, decides whether to approve or reject the new drug for marketing. So after checking all these checks, cross-checking, again reviewing documents, everything, they will come to a decision whether we have to release the drug or we have to ban the we don't have to permit the drug to release into the market. This ultimate decision will be taken by regulatory authority once you are completing phase 3 clinical trial, once you are submitting the new drug application. Next, even after approval, regulatory authority will continue to monitor the promotion of the product as well as for the information regarding any unusual side effects. That is pharmacovigilance, active pharmacovigilance. I said no. The sponsor company as well as regulatory authority will constantly monitor the drug. There is another pharmacovigilance also there, that is called spontaneous or unsolicited or voluntary reporting. Besides of that, we have active measures should be taken. That is active pharmacovigilance or solicited pharmacovigilance, which will be taken care by regulatory authority and sponsor company. Even though regulatory authority will giving permission to and approval to release the drug into the market, after release of the drug into the market, the regulatory authority will constantly monitor the drug, because during clinical trial, up to only maximum count will be 10,000. So but once the drug released into the market, that it will goes on for various kinds of population. There will be different geographies, different races, different hereditary conditions, different chemical environments, different body conditions, different posology, and so many factors will come into picture. So maybe based on all these conditions, the human body react in a different manner. Maybe the adverse drug reactions which were not seen or observed during the clinical trial we may have chance to get at the time of post-marketing surveillance, that is after release of the drug into the market. So for that reason, that unusual side effects monitoring, the regulatory authority will be constantly working, and it also gives some suggestions, and it will ask the sponsor company also to monitor proactively the drug and it's about efficacy and the side effects, that is adverse drug reactions. This is what happens in post-marketing surveillance. So that is phase 4, post-marketing surveillance or phase 4 clinical trials. This phase starts when the drug has been approved for marketing. Once the drug is reviewed for its information through NDA, then the consent sponsor company will get patents to release the drug into the market. So the phase starts when the drug has been approved for the marketing. This is a very important phase because it often garners additional data about the safety and efficacy of a product when it is marketed for patient use. Why we have to be monitored again and again? Because it will be exposed to more the number of subjects, the more number of patients. So there is very important phase. Even though you are proving safety information in the clinical trial, you have to be regularly monitored. That is why we were here, the pharmacovigilance people. We were continuously working on drug safety information. By that, we will contribute to the patient safeguarding health, patient safety. So the department existing in this health science, so-called pharmacovigilance, is concerned with the safety information, because before this pharmacovigilance, you have seen in the last sessions I've explained, with the simple chloroform, there were deaths. With simple diethylene glycol, there are 107 lethal cases. With thalidomide, which is the anti-sickness drug, that is morning sickness, and which is very usual drug. With that also, we have 4,000 limb deformities. So if there is no proper system of collecting the safety information, this areas will goes like a trend and will diminish the public health. So for that reason, this pharmacovigilance will be actively or passively, continuously monitoring even in the phase 4 and submitting the information. By that information, the FDA will reviewing again that information submitted by PV. So then it will come to decision whether it should ban the drug or approve the drug. So the sponsor company should not satisfy here. Yeah, I got this patents and I have to do whatever I can on a liberal way. That is not the thing. So even though you are getting patents, the regulatory authority will be monitoring your drug. If your drug is really causing some death or some serious drug ADR, then there is again chance of banning the drug. So you should be constantly monitor the drug, and you have to prove the safety again and again until unless it is having ultimate effects without any side effects. Next, phase 4 studies may be mandated by regulatory authorities or may be undertaken by the sponsor company. Not maybe, they are must and should basis, have to be volunteered by, monitored by sponsor as well as regulatory authority. This is to detect any rare or long-term adverse effects over much larger patient population. That is what I'm telling about all the things. Because during clinical trial, there will be limited subjects. So once the drug released into the market, there will be more and more number of subjects. So there may be chance of rare and long-term adverse effects. So that reason we have to constantly monitor again and again. Such adverse effects detected by phase 4 trials may result in the withdrawal or restriction of the drug. So if the adverse drug reaction will be a trend and which is very severe and which is meeting the seriousness criteria of death and life threatening, then there will be chances for drug withdrawal or restriction of the drug. Here I've just provided the summary of the session, that is what about the phase 1, phase 2, phase 3 and phase 4. Okay, phase 1 is safety. What is the information we'll get from phase 1? Only safety information. These are also called non-therapeutic trials or human pharmacology trials. What is the number of subjects involved here? 20 to 80 subjects. Next, we'll be giving this drug to only healthy volunteers in the phase 1 trial, and we'll come to know the safe dosage. How? We'll be testing the dose on escalated manner and increased ascending dose manner, that is single ascending and multiple ascending. We'll come to know maximum tolerated dose, and the maximum tolerated dose will be fixed. That dosage will be considered as safe dosage. Then we come to know what is the route of administration better suitable, like by that we can fix the formulation, the route through which we have to administer. Next we can come to know dynamics and kinetics. All this information will be come to know from phase 1. Phase 2 is about safety and efficacy information. So these are all called therapeutic trials. The number of subjects in the phase 2 is 100 to 300 patients. So here the patients, why you're calling? Because these are diseased candidates. Here we are calling just it as a healthy volunteers, but together they will be subjects, whether it may be patients or healthy volunteers. So here also we'll get safety as well as efficacy because we are giving the drug or patients. Next, as well like phase 1, we'll get kinetic and dynamic information. Phase 3 is an extension of phase 2 where we can confirm the therapeutic action. That's why this is called therapeutic confirmatory trials. The subject count will be increased from 300 to 3000 here. There will be comparison with placebo or active. What is a placebo? Anybody? What is mean by placebo?
Empty, empty drug.
Yes, inert material.
At the
Okay.
material.
Yeah. So we'll compare the drug activities and actions, therapeutic actions with the inert material or active worldwide, that is already widely marketing drug or the standard drug. So with the drug also we'll be comparing. Pharmacovigilance next. After phase 1, 2, 3 will be submitting documents so called new drug application. So the after new drug application, FDA will review and approve the drug to release into the market. Then will be releasing the drug into the market. This pharmacovigilance will work in both CT, clinical trial, and PMS. What is the process of this pharmacovigilance? This collect the adverse events. This will process. We will already discussed the workflow of adverse events, right? So that is a process will be happening, and that will submit to FDA. So even after getting patents, after phase 3 trial, so when we are submitting the information, AE information, safety information, then there is also chance for withdrawal of the drug or retention of the drug. Next, there are two types of pharmacovigilance. What is active pharmacovigilance and passive pharmacovigilance? In the last sessions only I have discussed. What is meant by active pharmacovigilance?
Sponsor company will take the follow-up about the adverse events.
Good. Sponsor company will be taking active measures and follow up for the patient records or directly ask the patients, collect the information, submit to FDA on regular intervals, regular dates. So for that reason, they will be getting paid. Okay. The sponsor company will pay to that amount to that people who are reporting that ADR. That is active pharmacovigilance. Next, what is meant by passive?
Voluntary reporting. They
Yes,
on their
on their
own they
own.
will.
Yes, on their own wish, they will be reporting the ADR information. And this passive pharmacovigilance, what this will do is only they will be giving some motivation to healthcare professional or consumers, patients. Like if you are reporting, it will be good sign like that. That's it. They're not getting any payment for that. So this is will be called as passive, so spontaneous or
Spontaneous.
voluntary reporting or unsolicited reporting. So there are four names for the passive reporting. Next, active pharmacovigilance will also be called as solicited reporting. So we'll be having another sources like literature articles, facts, email, internet, telephone, social media. We have so many sources where we get ADR information. So all this will contribute to safety, 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 safety. So the ultimate goal, because these days we were completely relying on chemical environment for simple nail pain and simple headache to the surgery. Post surgery will be giving anesthesia, right? So everything will be a chemical moiety and we were injecting into our body. So it is fine because we were living in such a chemical environment, but whatever the things, whatever the substances you are taking into the body, it should be safer. Otherwise we will again suffer with some other abnormal body conditions. This is all about the beautiful concept so called clinical trial, which is very very important. And is it any tough point here? Any queries? If you have any queries at any cost, you can ask now. Team? You can ask now. Any questions? Okay, I will only ask. So after preclinical trial, what is how the information will be sending to FDA? So what is the format? What is the document you will be sending after preclinical trial, not clinical trial? Team, am I audible?
INDA.
That is investigational new drug application. So after clinical trial, phase 3 clinical trial, what is the document you'll be sending to FDA to approve, to get approval? Yes. After phase 3 clinical trial, what is the document you'll be sending to FDA?
New drug application.
New drug application.
What is the number of subjects involved in phase 1?
20 to 80
20 to 80.
voluntary. Healthy
Yes.
voluntaries.
Next, what is the number of subjects involved in phase 2?
100 to
100
300
to 300.
patients.
Next, phase 3. Phase 3.
1,000 to 3,000.
Yes. So what is the difference between phase 1, 2, and 3?
Phase 1 is about only safety.
Yes.
Phase 2 and phase 3 is about safety and efficacy. To
What is the
determine
difference
safety.
between phase 2 and phase 3?
Number of volunteers.
Number of
Therapeutic
subjects.
confirmatory trials.
Good. So both are therapeutic trials, but here we'll be confirming the therapeutic information, so-called therapeutic confirmatory trials. Okay. Yes. Next. So in which phase you will be finding the maximum tolerable dose?
Phase 1.
Phase 1. Good. Next.
Single ascending. That.
Yes. Single ascending dose studies. Yes. That's it. If you have any doubts, you can ask me. Pause the session also. Otherwise, we can wind up the session.
Okay.
Bye, team. Bye, everyone.
Bye everyone.
Yes, Sonali, you can wind up the session.